Hi everyone, it's Mr. Vallejo. Welcome to biology class. Today we're going to take a look at human genetics. So let me go ahead and share my screen with you. Remember, these slides are available for your use in your learning management system, whether that is Schoology or it is Canvas. Your slides are available there. Human genetics. Now, when I was at UCLA, I took a whole entire course on human genetics today. Of course, we're not going to cover everything in that course, but we'll take a look at some interesting topics. Before we do that, we're going to review some basic terms that we know from our studies of mitosis and meiosis and genetics. So let's go ahead and take a look at these four sets of terms. We have a gene and an allele. A gene, as you know, is a DNA sequence that codes for a protein product. Um, So if you take a look at this chromosome, remember this is a duplicated chromosome with two chromatids, and the gene is represented right here. It says it's a segment of DNA that codes for a trait. It doesn't necessarily have a band on it, but that's what it's showing in this picture here. Sometimes there's a band and the gene is actually just below or just above it, and we can use the band for reference. An allele is a different form of the same gene. You can see I have some different eye colors there, but these eye colors are due to the same gene. You might just have different copies of different alleles. Those are different forms of the very same gene. There may be different alleles in the population, so that is how you get uh, all kinds of different colors in individual people. All right, I'll remind you of the term homologous chromosomes or homologs. And homologous chromosomes are the chromosomes that bear the two alleles for each characteristic. They are the chromosome pair. Okay, when we say that you have 46 chromosomes and 23 pairs, let's say you took chromosome number 5. Chromosome number 5, you have two copies of them, and this would be two homologs or two homologous chromosomes of that chromosome 5. On that chromosome, you have the same genes, and you might have the same allele at the same physical location or locus, like you have here. This is a homozygous dominant for this gene. This is homozygous recessive for this gene because you have two copies of this gene, one on each homolog. That's a recessive form. Now, in this case, this is where you have a heterozygote that has one of each of the two different types of alleles. One is dominant and one is recessive. Now, the term autosomal, it's for the first 22 chromosomes. Chromosomes 1 to 22 are the same in males and females. And so you can take a look at the karyotypes that are in this picture right here. And you can see that the only difference between the two karyotypes is that 23rd chromosome here. And if you take a look at 23rd chromosome, those are called the sex chromosomes. If you have a Y, you're a male. So this is the 23rd chromosomes for male. This is the homologs of the 23rd chromosomes for female. This is XX, this is XY. And Y is much smaller. The Y chromosome has gene for development of the testes. So if you have the Y, the testes develop, you're a male. If you don't have the Y, then the testes don't develop and the ovaries are allowed to develop because they're not getting that input from the genes that a code for a protein that is the signal for the development of the testes. Alrighty, I'll remind you as we studied meiosis, we learned about crossing over and different ways of producing new combinations of alleles to increase genetic variability. One way of increasing genetic variability is by increasing the percentage of genetic recombination. You can see here what's happening. These two homologs get so close together that they form right there, chiasma, and this tetrad formation with four different chromatids, they're so close together they swap information. So that's when you get novel mixtures of the genetic material. This increases genetic variability. This is called genetic recombination. That's one of many ways that genetic variability is increased in our body. For example, last time we went over the uh, the idea that out of 5 million sperm, only one of them wins, and so that unique combination of genes is represented in the offspring. Remember, it does depend on where the chromosomes line up during metaphase of meiosis. That seemingly random event actually has different outcomes depending on uh, where that chromosome lines up during metaphase in the equatorial plate. We know that some genes have different alleles, and you may carry different alleles on the same homologous chromosomes, and so you can be homozygous dominant, homozygous recessive, or heterozygous. So these are all ways to increase genetic variability, and crossing over is a way to increase genetic variability in in humans. And then the other term that we want to review with you is a karyotype. Um, New technologies can provide insight into one's genetic legacy and reproductive decisions. For an increasing number of genetic disorders, tests are available that can distinguish carriers of genetic disorders. And so here's the karyotype, and you can see the photograph um, of your chromosomes, and you can see that they've been matched up so that you can see that they're the correct number of chromosome pairs. The fetus that gave up this karyotype is also a male because you can see there's an XY for the 23rd chromosome for the sex chromosomes. Now the karyotype is comes to us because the fetal skin cells that are in the amniotic fluid are used and grown, and then we can examine them. We get this through amniocentesis, but we can also get it through CVS. That's chorionic villus sampling, which is a much less uh, intrusive procedure. As you can see in the picture here, you have for an amnio, what you need is you need to do a sonogram so that you can stick a large needle into the amniotic fluid and withdraw some of the fluid that way. You should be able to get some of the cells the same way as you go this way, and you can do it at a much earlier stage. Get those fetal cells right there from the villus, and so that's actually plural as the villi. So you can extract tissue from the villi. There's a little finger-like structures in the chorion, and then grow those, and in several hours you can get a karyotype. So there's a young lady who is, man, that is an old machine, but look at the results. I remember when my niece was born, her mother, my sister-in-law, sent out baby's first pictures, and this is what she sent out. So that is what you get when you do an ultrasound and get a picture of the fetus that way. Used to be that a technician would print this out on paper and then use scissors and cut them out and paste them. They could do it pretty rapidly. Within 5 minutes, you would have a karyotype. That's old school though. Now you just put that on a computer screen, add color to it, bam, you can match that up pretty quickly. I'm told it takes less than a minute to do that stuff. There's more information about karyotypes. Should you be interested, especially those of you who are allied health majors. Okay. As far as screening goes, this is an old picture from 2004 of the uh, newborn screenings available by state. California was actually pretty low, and so it used to be that you would prick the baby's heel, take a blood sample, and put that on this card. And then there are chemicals already in the card, and you could you would get an instant result. to check for the existence of different medical conditions. But as we'll see a little bit later in a different talk, just because we can do something doesn't mean we should do that something. There are all kinds of just different ethical issues that come about. Um, how about this one? Do you terminate a pregnancy if your baby has a birth defect? Um, hmm, that's a tough question. I know for many of you the answer no, but I have a friend who did this very thing. Her marriage was in trouble. She found out she was pregnant. She was over 40. The child was going to have Down syndrome, so they decided not to have their baby. I don't know if I would be able to do that, but that's my own choice. And so that's something to look out for. All right. Um, the main part of this talk has to do with gene locations on the same chromosome. That's called a linkage group. All the genes located on a given chromosome are inherited together, so that's why we call it a linkage group. Wherever gene P goes, gene L will go. Why? They're on the same chromosome. So they're on the same chromosome, they're going to be inherited together. All right, here's some sex-linked traits. This is hemophilia. Here's a picture of a person with hemophilia. Hemophilia is a disorder that has been studied for hundreds of years because it was seen in the royal families of England. And in the royal families of England, what you would have is people who would marry each other because they were both royalty, or if the kingdom of Vallejo was at war with the kingdom of the Granny Smiths, then I would offer my daughter, and they would offer the son, and they would have a marriage between the royal families, and that would create peace between those two families, those two kingdoms. So because of that, we know a lot of the history behind the marriage lines, and so we could study hemophilia as it went through the royal families. Um, Hemophilia disease, if you used to be if you got a cut, you would bleed out and bleed to death. There are now chemicals, medications, stuff to help people who have that, but it's still certainly a different lifestyle. Red-green color blindness is also a sex-linked trait. Remember, a sex-linked trait is a trait that does not necessarily have to do with the name implies that it has to do with the 23rd chromosome. Okay, so what it is that a woman has XX and the gene is on the X chromosome, but a male has XY and the Y has virtually nothing on it, much smaller, and it has fewer genes. So the Y doesn't have the gene that the X has. So it's more likely that a male has a disorder because the male doesn't have another chromosome with a different dominant genotype that's going to wipe out the disease-causing allele. So if you can see the number 16 right here, what you have is normal vision, normal color vision. But if you can see a different number, I don't even know what it is for this one, then you might have red-green color blindness. Some people, instead of seeing red and green, they see various shades of gray when it comes to red and green. So that is a characteristic that has to do with the uh, sex-linked traits. And then male pattern baldness we'll get into a little bit later, but we'll see that if you're male and your grandpa on your mom's side is bald, then there's a really good chance that you're going to be bald too. All right. In order to do well on the coming quiz, you need to know that Morgan worked with fruit flies, the fruit fly named Drosophila, in order to do any mapping. Mapping is the process where if you take a look at genes on a chromosome, you can see how far they are apart relative to each other. So over here, um, here's three different hypothetical genes, genes G, C, and L. You can figure out uh, how close they are together right here by measuring the amount of recombination crossing over between those genes. We do that experimentally. We have these numbers here, this data. From this data, you can reason out that G and L have much more crossing over, so they're far apart because there's more distance for the genes uh, to have crossing over events occur between them. But if two genes are close together, like G and C here, they're closer together, it's going to be fewer crossing over events. Similarly, C and L are going to have fewer crossing over events, but between G and L, you might have twice as many. And so that's how we know that C is between G and L. So this certainly could be instead LCG because they're relative to each other. And so when we figure that out, that's how we do that. All right, to finish up today's talk, we're going to take a look at some processes that happen. We have in humans, we're going to see, take a look at Tay-Sachs, achondroplastic dwarfism, male pattern baldness, and some disorders of the chromosome numbers in the sex chromosomes. Now you can figure out many things from a family tree or a pedigree. And when we do a pedigree, we typically try to figure out not these types of characteristics, which are fun to look at, and you'll do that in the lab this week. But more importantly, you can take a look at how diseases are inherited. And you can see in this one here, this is a pedigree of a family that has deafness running through it. And so Jonathan Lambert is deaf, and he got that from, looks like he got a big D from, must be, oh, they got a little d from each his mother and his father, and that's why he used little d, which showed the recessive deafness trait. And since his wife was heterozygous, who was not deaf, but she was a carrier for that deafness, statistically, half their kids should have had deafness, and 2 out of 7 did. So that's how you read that. Many inherited disorders in humans are controlled by a single gene. That's not true for other things like intelligence, like hair and eye color. There are multiple genes or the multigenic or polygenic traits. But um, some of the disorders are single gene disorders, and you can see in this list some that you might recognize: cystic fibrosis, sickle cell anemia, Tay-Sachs disease. Those are recessive disorders. Um, Here's a dominant one we're going to see in just a moment. It's achondroplastic dwarfism. And I have this one, hypercholesteremia. So most human genetic disorders are recessive, so you only have a 1 out of 4 chance if both parents are carriers. But that's what happens here in Tay-Sachs. If you have two people who are carriers, they can have a baby who will have a limited lifespan. And so a lot of text on this slide, but basically what happens is there's some chemicals that build up in the brain, and then the nerve cells get surrounded by some fatty tissue, which causes neurological problems. Um, Very rare disease, but more likely in the uh, Ashkenazi Jewish population of Eastern Europe. So you have two people who have that background, and you should get tested. Um, Even if you get tested and you are both carriers, you still have only a 1 out of 4 chance of having a baby who has Tay-Sachs, but then you'll have the information to know whether you should just avoid having a baby. Maybe you can adopt a baby. maybe you have the baby anyway, knowing that there's a 75% chance that there will not be a problem. When I was a student at UCLA, I was tested for Tay-Sachs. I was walking up Bruin Walk, and the Jewish Student Union had a table, and some of their members said, "Hey, you want to get tested for Tay-Sachs?" And I said, "Yes." I was a freshman. I didn't know what that was. So I got tested for Tay-Sachs. Turns out I'm not a carrier. So even if my wife were a carrier, she's not. Um, and we would have no chance of having a baby with Tay-Sachs. All right. Here's some of my friends who are shorter than I am. They are little people. Don't call them dwarfs. They don't like that. They're called little people. And so my friend Evan and Michelle was actually in my college class as a student. Um, my friend Marilyn is the matriarch to this family. And then Dan is my friend who works he's a manager at a local grocery store. They have an older son whose name is Clint, who has a short girlfriend, not a dwarf girlfriend, but a short girlfriend, much shorter than him. Clint is like 6'2", so he has a normal height. Uh, here's Evan and uh, and his daughter Michelle. Back in the background, my daughter right there. This is an old picture. This must have been a church. It looks like after service or after vacation Bible school from those shirts there. Here are Dan's kids. Um, they're Trent and Autumn, who are both little people as well. Here's me and Evan. Evan used to kid with me and say, "Hey, I'm naturally taller than you are. We sit down." Because people with this type of dwarfism, achondroplastic dwarfism, their limbs are shorter, but their torsos are not affected. Is Evan taller than I am when we sit up? Possibly. Sex-linked recessive traits, sex-linked genes. Remember, these are the sex chromosomes with the on the X. I remind you that the Y doesn't carry anything. Um, So this was discovered by Morgan, and again, they did this in the fruit fly. And here are some Punnett squares that you can take a look at so that you can review how the the traits are inherited in sex-linked traits. Remember, the Y doesn't carry anything. So because the Y doesn't carry anything, a male is more likely to have the issue, as you see right here, because although this X has a recessive characteristic which causes the condition, the male does not have another X with a big R to cancel that out. So that is why we have more males with it. Males can't be carriers because they only have one. They either show it or they don't show it. A carrier is a female who doesn't have the condition but carries the gene for that condition. So again, hemophilia and red-green color blindness are sex-linked recessive traits that I already went over just a little bit. And this we're going to do a little bit later in the genetics problems video. So we're going to skip over that for now. But here's a guy with male pattern baldness. Remember, I mentioned if your grandpa on your mom's side was bald, then you're going to be bald. Why? Because let's follow that. Grandpa was XY. That means he gave his X with the recessive gene to your mom. And then your dad gave a Y and your mom gave you an X. So you have a chance that your mom is going to give you the X with the recessive gene on it. So sex-linked recessive traits like male pattern baldness are inherited this way. All right, as we finish up, we're going to take a look at how this all happens in meiosis. Genes are located on chromosomes whose behavior during meiosis and fertilization accounts for Mendel's inheritance patterns. Now, we've studied this before, and this is meiosis. As you can see, there's metaphase and anaphase. This is meiosis 1 right here. There's meiosis 2 down here. But the thing is, if you have an accident during meiosis, then the chromosome number can be affected. Now, these things down here, asbestos, radiation, ultraviolet light, these are things that can affect whether your chromosome number is changed. They can affect whether the chromosomes separate correctly during meiosis. So these things down here are called mutagens because they cause mutations, and a mutation is a change in the DNA. So with non-disjunction, what it is you're going to have an abnormal chromosome count, possibly, if you have a failure of homologous pairs to separate during meiosis 1, like right here. This is not typical. One should go to the right. And so these guys right here are going to have 3 copies, and these guys over here are going to have 1 copy. So they're going to have the wrong number of chromosomes. Over here, it happens in meiosis 2, and you're going to have some that have the normal number, but the other half of them are going to have an abnormal number of chromosomes. So if you take a look at what happens in fertilization, when you have an egg and a sperm coming together, and they have too many copies of a homologous chromosome, then that's where you get 3 copies like you do right here of this larger one. A common one that you may be aware of is trisomy 21. Trisomy 21 is when you have an extra copy of the 21st chromosome. And so a person with trisomy 21 has certain physical characteristics that we expect. And so you probably know or met or have encountered someone with Down syndrome. So that is a chromosomal aberration. We have too many copies of chromosome 21. It's more likely to happen if the mother is, excuse me, if the mother is older. And so typically, if the mother is over age 35, they'll offer an amniocentesis, so you can run a karyotype to see if the fetus has 3 copies of chromosome 21. Now you can also see that you can have an abnormal number of sex chromosomes. Rule of thumb, if you have a Y, you're a male. If you don't have any Ys, you're a female. So let's take a look at it. Here's the first one. This is Turner syndrome. And Turner syndrome occurs when you have not 46 but 45 chromosomes. A person with Turner syndrome has these characteristics. They have a short stature, ovarian failure, webbed neck, and a shield chest. And you can see in the karyotype right here, there's nothing there, it's missing. So this is a female, she has an X. So that's why up here it says XO, because there's no second chromosome. I know someone with Turner syndrome, and she is shorter, and she's able to actually, she can pull the skin up on her neck a few inches, which is fun at parties, I imagine. So Turner syndrome. Klinefelter syndrome is when you have a male with an extra X chromosome. He's infertile, has some breast tissue development, has some secondary female characteristics, taller than average. The person with Klinefelter syndrome. This is the male, but he has some female characteristics, as you can see. And there's a lot of text on the slide, but psychologists used to teach us about the super male syndrome. which is when a male has two Ys. And if you don't want to read the whole text of this slide, you can see that people with two Ys are typically have lower intelligence, they're over-aggressive, it's unusual, and you can see that over here that less than, it's a very low percentage. Out of 1,500 karyotypes, they only found one person who was XYY in a population, looks like in the UK. And they're typically taller than usual. Super male syndrome. An abnormal number of sex chromosomes, it's certainly something different, but it doesn't usually affect your survival. You can have various degrees of malfunction. My friend who has Turner syndrome is actually not infertile. She's been told that she can have a baby. So varying degrees of malfunction, you can see that. Instead of having a different number of chromosomes, you could have the same number of chromosomes in your karyotype, but you could have still a problem. Here are 4 different ways you can have the problem. You can have a part of a chromosome deleted. You can have a part that is duplicated more than once. So that's a deletion and duplication. An inversion happens when you have 2 adjacent genes that they're still there, but they flipped. And and then you also have a situation where you can have that gene, that piece being cut off right there, and then that is going to be swapped out with a piece from a chromosome that's a totally different number, a non-homologous chromosome, and that's called a translocation. So careful, even though you have the right number of chromosomes, you may still have an issue. This particular issue, this type of translocation has been shown to be the reason for this type of cancer. It is a gene that is activated when this happens, called the Philadelphia chromosome. You can see in this old school picture, this one's shorter and this one's longer. And so what happened was a piece of the chromosome from chromosome 9 was taken and put on the smaller one over here. That piece right there is taken off and put over here, and that activates that gene, and that gene happens to cause cancer. So that is not what you want. All right, in order to do the quiz correctly this week, I want to remind you of the situation where you have universal donors and recipients. A person who can receive blood from anyone is a person with O-type blood. And no. To receive from anyone is a person with AB blood. You can see there's no clumping going that way. And a person with O-type blood can give to anyone, called a universal donor, and their blood can be received safely from any O can give to any other blood type without the risk of clotting. So that's what you see in this diagram. That's just a review of something we saw last time, but I know it's on the quiz. All right. So those were some topics in human genetics, and so there's all kinds of different topics. This is a very interesting topic. And I remember hearing about all kinds of different things in my human genetics class. That's just one lecture, just one short lecture. And so if this is interesting to you, I encourage you to seek out more information, do your own searches, take a human genetics course when you get to the big U. And I hope that I piqued your interest a little bit today in the area of human genetics. I'm Mr. Vallejo. Thanks for coming to biology class. We'll see you next time. Bye-bye.